Okay, Banu is doing her master's at ETH, and she's going to talk about TargetCall, which introduces the idea of pre-basecalling filtering. And this work was also presented in an earlier session at RecomSeek, so she's going to repeat the talk and do it even better.
I hope so.
What else?
Set up the microphone.
Okay, you need to set up the microphone, that's right. Microphones, since we have two setups. And we're running 15 plus minutes late, unfortunately.
Do you have an idea how to
Banu is
make
going
up
to
for
speak
that?
fast. Are you going to speak fast, Banu?
I can. So I think it's a little longer than
Make it exciting. And then the lesson is done now.
I'm kidding again.
Yeah. We can leave Danna's talk then.
I'll just bring everyone coffee and we can skip the microphone.
Yeah. Yeah, we started late, but yeah, Danna put us later, and then I asked people to ask questions to Joel. So.
I also didn't shorten that introduction as much as
That's true. You did that.
Okay, I think we should get started without more delay. Yeah,
Okay,
go ahead.
What?
She wants to take a perfect picture of you with your name over there.
Okay. As Onur said, I'm Banu and I will present TargetCall, which was done at ETH Zurich by the Safari Research Group. Okay. And our motivation in this work is that basecalling can consume up to 84.2% of the total execution time, and it becomes a major computational overhead for the pipeline. And what we observe is that much of this computation is wasted because majority of the basecall reads are discarded after basecalling. And our goal is to eliminate this wasted computation in basecalling with high accuracy, scalability, and adaptability. For this, we have the idea of proposing a pre-basecalling filter that would filter out the user's reads before basecalling. And we propose TargetCall as a new pre-basecalling filter that achieves this task in 2 steps. First, it uses a LightCall, which is like lightweight basecaller, to perform noisy basecalling on the raw electrical signals. Then it compares these noisy electrical signals to the reference genome to predict if the read is going to be useful. Our evaluation shows that we can improve the basecalling end-to-end basecalling performance by 3.3x by filtering out majority of the user's reads. And when we compared it against the targeted sequencing approaches, we show that it achieves better recall, precision, throughput, and performance than the prior targeted sequencing approaches that are repurposed as pre-basecalling filters. Let's start with background and motivation. Genome sequencing enables us to determine the content of one organism's genome, and it's important for areas such as in precision medicine, outbreak tracing, and understanding of evolution. And nanopore sequencing is a specific sequencing technology that we can use for genome sequencing that's widely used and that produces long reads of tens of kilobase pair length with high throughput and low cost. And the traditional pipeline for nanopore sequence analysis starts with genome sequencing, where the DNA fragments are converted to raw electrical signals when they are passing through the pore. Then these raw electrical signals are converted to a set of bases called as reads during basecalling. And the common next step is to perform read mapping, which is explained a lot in this presentation already, where we match the read to the reference genome to determine if the read is going to be mapped or unmapped. And prior studies shows that just basecalling step takes up to 84.2% of the total execution time, whereas the remaining downstream analysis takes less than 16%. And we conclude that basecalling is really a major computational overhead for the traditional nanopore sequence analysis pipeline. And our observation in this work is that what worsens this situation for many applications is majority of this computation is wasted because we discard many reads after basecalling. One such application is SARS-CoV-2 genome assembly, where less than 1% of the reads are useful, meaning not discarded after basecalling, even though the entire dataset is processed fully during basecalling. And the typical reason why you have 99% of the reads as users is that they do not match the reference genome that is of interest by the application you are targeting. So the key takeaway is the user's reads waste significant amount of basecalling computation in the traditional pipeline. And we have an alternative for this, which is targeted sequencing. Instead of traditional pipeline, where we sequence all the reads and send them to the next step, in targeted sequencing, we perform sequencing selectively only on the useful reads. This is enabled by a functionality of the nanopore sequencer, where you can stop sequencing some certain reads with a reject command. And in targeted sequencing, the rejected DNA fragments are discarded from the sequencer, and the accepted ones are fully sequenced and sent to the base calling step. But this requires targeted sequencing to identify or have a method that would identify which reads are useful with reliable and high accuracy. And we have identified some limitations of these methods. They are not really intrinsic limitations of targeted sequencing, but we identified that all of such methods that perform targeted sequencing suffer from at least one of these three things. First, they have low sensitivity and they falsely reject many useful reads. Or they have poor scalability in the sense that their performance and accuracy will drop significantly with increasing reference genome sizes. Or they have lack of adaptability to different use cases, so you cannot use all of them as is for any use case that you have. You might need another preprocessing step. And this is because many of them use, or some of them use, neural network classifiers to classify if the read is useful and uses, and this classifier needs to be trained for each read set and reference genome we have. And that's why targeted sequencing approaches are not robust solutions for eliminating the wasted computation in base calling. And our goal is to eliminate the wasted computation, but unlike other approaches, we want to maintain high sensitivity in keeping useful reads. We want to be scalable to large reference genomes, and we want to maintain adaptability to different use cases. For this, we have TargetCall, which is a pre-basecalling filter. And we need to explain what's a pre-basecalling filter, but before that, let's remember our approaches. Our first approach is traditional pipeline sequence and base call everything. Our second approach is targeted sequencing, where we selectively sequence some things, and we show some limitations of these approaches. And what we propose is pre-base calling filtering that performs base calling selectively on the useful reads. And this is different from traditional pipeline because we don't base call everything, and this is different from targeted sequencing because we perform sequencing fully, but we perform selection on the base calling step. And it's the opposite in actually targeted sequencing, where you do selectively sequence things and you base call everything that is sequenced by the filter. And when we do that, we are actually converting the problem to an offline problem, where you're just starting your filtering after you are completely done with sequencing, and you have the full signal data coming from the sequencer to be able to classify your read. And what we do is we just at the filter step between sequencing and base calling step, and this filter needs to reliably identify whether a read is useful and uses using the entire raw signal. And then the filter should stop the analysis of useless reads and should forward the useful reads to the base calling step. So the high-level goal of TargetCall is to really predict if the read is going to be useful for base calling. And it does so with two steps. First, LightCall completely base calls the raw signal that is coming from the nanopore sequencing in a noisy way. And then Similarity Check compares this noisy read to the reference genome we have. And if the read is useless, the analysis of that read is stopped at this point. Otherwise, the read is forwarded to the base caller so that we maintain the base calling accuracy for the useful reads fully. And TargetCall has high sensitivity because we use a highly accurate Similarity Check component, has good scalability because the performance of LightCall model is independent of the reference genome that we have, or the size of the reference genome we have, and is adaptable to any use case without any changes because LightCall model is generic across use cases and it doesn't require any retraining of the neural network model that we have. And that's why TargetCall is a robust solution for eliminating the wasted computation in base calling. Let's check our components, and our first component is LightCall, which is a lightweight base caller with a simpler neural network model that performs base calling in a noisy way. These reads are still highly accurate for us to be able to perform filtering, but not accurate enough, not as accurate as we would have in full base calling. And we designed LightCall architecture by simplifying the state-of-the-art base caller Bonito, and we performed the simplification in a way that we improve the performance significantly while maintaining most of the accuracy benefits of the complex neural network model we have. And we use 3 steps for doing this. First, we reduce the channel size of the convolution layers. Second, we remove the skip connections. And finally, we reduce the number of basic convolutional blocks we have in the model. And the final LightCall architecture is a series of, or like a repeated convolutional blocks with where each one has one or two units that include grouped convolution, pointwise convolution, Bayesian normalization, and riddle layers. And the second component is similarity check that tries to find if a noisy read coming from LightCall and the reference genome that is determined based on the application that you are targeting is similar to each other. And depending on the answer, it labels the read as useful or useless. And this problem is very similar to read mapping problem, and we use the state-of-the-art read mapper min-map2 for this problem. And when we show that if we combine LightCall and similarity check, so if we use the noise reads coming from LightCall module in the min-map2 implementation, we can still get very high sensitivity, up to 99.45% for keeping useful reads. And we show later in the paper that this is good enough because the loss of 0.5% of the useful reads can be tolerated with high sequencing depth of coverage. We have three use cases where we show that TargetCall is really scalable to large reference genomes and adaptable to any use case. The first one is SARS-CoV-2 detection, where we have the COVID as the reference genome that we have as a small reference genome. And we are trying to identify the COVID reads among the COVID and human sample, so that basically anything that is similar to our reference genome is labeled as useful for this use case. And we have a similar use case where we have bacterial and viral samples where we try to identify the viral reads. But this time our reference genome contains multiple viral species, so it's a complex reference genome compared to the first one. And the last use case is targeting sepsis detection, where we have a large reference genome to show that we can actually really scale TargetCall to large reference genomes well. And this use case is different because in this use case our sample has bacterial and human reads, but we want to base call every bacterial thing. Since we don't know what you have as the bacteria in your sample, we just deplete every human things, and that's why in this use case, if your read is similar to the reference genome, you label that as useless instead of useful. This is very different than the other ones, and this is going to be important for the results. To show the benefits of TargetCall, we have two experiments. The first experiment is to show how much base calling speed up we can get with TargetCall, and we do so by comparing Bonito alone with Bonito preceded by TargetCall to eliminate to filter out the user's reads before base calling. And next we compare TargetCall against targeted sequencing approaches uncalled and SIGMAP. And our methodology was to use the labeling mechanisms of these targeted sequencing approaches as pre-basecalling filters and applying full base calling on the useful reads. And we compared the execution time, recall, and precision of these tools. Our data sets contained a lot of things. Read sets, 5 different read sets from various organisms. 4 of them are real and sampled from prior work. 1 of them simulated using Deep Simulator. But we open source everything, so our results can be reproduced. And our reference genomes, we have 4 of them with various reference genome lengths and ratio of useful reads in our samples. We used a system with NVIDIA 100 and Titan V GPUs for the LightCall component of TargetCall, and an AMD CPU with 0.2 terabyte main memory for the similarity check component. But for SIGMAP and uncalled, we have a system with 1 terabyte main memory because SIGMAP and uncalled requires more memory than we have in the first system for large reference genomes. And our first result is to compare or to show how much base calling speed up we can get with TargetCall. And the TargetCall execution time includes both the filtering time and the base calling time of the useful reads that are identified by TargetCall, not the ground truth ones. And we show that on average we can achieve 3.3x base calling speed up with TargetCall. And if you compare the performance of different tools or different targeted sequencing tools with TargetCall, we see that on average, TargetCall performs up to 9.7x better than prior tools. And more importantly, this speed up is actually, the performance improvement is more significant for large reference genomes, where we show that TargetCall is 13.3x better than SIGMAP for the large reference genome. And we cannot even compare it to uncalled because uncalled cannot even generate the index for the human reference genome we have in this specific configuration, even though we have a system with 1 terabyte main memory. And we got a memory error during indexing for this tool. Next, we compared the recall, which refers to the ratio of useful reads that pass the filter among all the useful reads. And we show that TargetCall achieves more than 99% recall on average, And TargetCall can achieve up to 23% better recall than prior tools on average. Similar to performance, the recall improvements are going to be more significant for larger reference genomes as well, where on average we are 15% better than the prior tools. But one thing that is important is that sepsis use case doesn't really measure the recall for finding human reads in the human sample. And we were curious if we really apply for a use case uncalled and SIGMAP as well as TargetCall, where we have a large reference genome, and what would be the recall for finding reads that are coming from that large reference genome. And we measured the recall of these tools for finding human reads in a sample, and recalled and SIGMAP and uncalled only had a recall between 40 to 53%, where uncalled showed more than 96% recall. And our conclusion was that TargetCall consistently provides high recall for all reference genome sizes that we tested and for all types of experiments we tested. Finally, we compared the precisions of these tools, and here precision refers to the ratio of useful reads that pass among the all reads that pass the filter. And on average, our precision is more than 92%. And it's significantly better than prior tools, approximately 60% better on average than prior tools. We have more details in the paper about targeted sequencing, different LightCall designs we have, and more details on our evaluation methodology, more evaluation results such as the basecalling speedup and recall and precision provided by different LightCall architectures, and how we finalize the TargetCall design, and to an accuracy and performance analysis across the entire genome sequence analysis pipeline, including variant calling, our throughput comparison to SIGMAP and uncalled, where we have more than 1100x better throughput than these tools, which suggests that you can actually use TargetCall as a real-time analysis tool as well, and the peak memory comparison of TargetCall against that of SIGMAP and uncalled. Please check our paper for more details, and we also open source our implementation, our evaluation scripts, and our data sets in our GitHub page. Let me conclude. So we propose TargetCall, which is an accurate, scalable, and adaptable pre-basecalling filter that works in 2 steps. First, it uses a lightweight basecaller to perform noisy basecalling on the raw electrical signals, and then it computes the similarity of the read to the reference genome to predict if the read is going to be useful for basecalling. Our evaluations show that we can achieve significant basecalling speedups by filtering out majority of the useless reads with TargetCall with high sensitivity. And we show that if you repurpose targeted sequencing approaches as pre-basecalling filters, TargetCall achieves better recall, precision, performance, and throughput than these tools. Okay. I guess I'm fast enough, and do you have any questions?
Okay, great. Thanks, Banu. Any questions? Okay, one is a question. Maybe fastest.
I have a question. So you explained what are the issues with the targeted sequencing, right? But it's kind of based on a similar tool, right? Because they are also using neural network there. So my question is, after you have trained your pre-basecalling filter, how is it? LightCall.
Yes.
Could you use it inside the sequencing machine to get rid of those?
Yes, you can use it, but the difference is that in targeted sequencing, you are doing this problem online. So you are trying to determine if the read is going to be useful while the read is being sequenced, so you don't have the raw electrical signal. And that's why you need to tune your similarity check component such that it only checks the initial parts, and it uses a noisy read that is coming from LightCall and tries to predict in that way. And doing that accurately is harder than doing the pre-basecalling filtering accurately. But in terms of throughput, yes, LightCall matches the throughput of and actually exceeds significantly the throughput of the sequencing machine. So there is no reason why, if you have a correct similarity check module, there's no reason why you cannot use LightCall for real-time analysis.
But that would, in summary, would save time, right? Also from the...
Exactly. From the...
Try that.
I didn't try that, but we can try.
You can try? Okay.
Yeah. Because that is really different. So you cannot, you can try it as is as well, but it will have the limitations of prior tools as well. I don't think we would be able to achieve very high sensitivity in keeping useful reads, as high as we achieve now for the pre-basecalling filtering problem. But as I said, we could optimize the module for this particular problem.
Okay, sounds good. Thank you.
So you gave the sepsis example, and From my understanding, the reason that the other tools have such low performance for the sepsis case is because you actually require the precision to be higher in that case. Am I right?
Exactly.
Okay, so what makes those tools? Then again, what makes TargetCall so much better at precision than those tools in all cases?
Okay, so basically, I think the answer is different for the tools that we compare. For SIGMAP, for example, they have a very complex index that uses KD trees, and it's like the tree size becomes very huge as you increase, and also the depth and the number of branches you have becomes very huge as you increase the reference genome size, and you have to traverse all of them. And what they do is they never convert the raw signals to bases, and they have to do approximate matching of the signal values because nanopore sequencing doesn't produce exact raw signal analysis. So that's why they have to match these exact floating point numbers with a very complex tree, and their performance drops significantly for filtering time, and their precision is also not good because they, I think, limit the tree length or the tree size somehow, so that you have much more like candidate locations or so, and then they perform.
They check just like one part, and then they don't check the rest, so they.
Exactly, or maybe they just say, oh, it's possible, we just say it as useful, and then their precision is not that high. For uncode, it's actually different. It's actually still quite fast because their index structure is optimized. The filtering time is fast, but then what they do is, so once they have the raw signal, they try to generate many possible match locations. And when your reference is large, the many of them are like too much. And even though they are not matching in the base pair, it's the likelihood of matching them in the raw signal domain is very high, so you have many more options. And because your reference genome size is large, and they don't want to lose recall, so that's why their precision is too low. But in TargetCall, what we do is we do base calling, but we just do it fast. And our accuracy is still good enough. So, for example, for the most complex model we tested, it's 16x smaller than the basecaller that we used to optimize LightCall, but the basecalling accuracy is not reduced more than 4%. And that's why we can still be very high precision and recall.
So, can we say that TargetCall is currently the only tool doing what it does?
Pre-basecalling filter.
Yeah.
So for the pre-basecalling filter, yes, and it's actually the first tool that says it explicitly. But there are neural network-based classifiers, but they don't do basecalling, so they just classify the reads. So people have used neural networks for this type of problems, for targeted sequencing, but I don't know any basecaller that is designed specifically for being fast by trading off some accuracy. I mean, there are basecallers that doesn't have that much accuracy, but it's because they are very old. Okay.
Okay. Any other questions? Otherwise, I think we're done.
Okay.
Thanks, Banu.
Thank you.